Why don't we get started? So welcome everybody. I'm Tim Block, and I actually see I'm wearing the same jacket in this picture that I am here. I do have another jacket, by the way, and tie, but this is the Hepatitis B tie. I'm the president and co-founder of the Hepatitis B Foundation. That's Baruch S. Blumberg Institute, and this is our AASLD reception. And normally we'd be greeting you all, having wine, or in this case, club soda, and I really look forward to these events at the AASLD meeting, where the foundation welcomes its friends, and everyone in the hepatitis B community gets a chance to spend a couple of informal hours with each other, and gives us a chance to tell you a little bit about ourselves and what we've done every year. But so I really thank you all for coming to listen to us, despite the fact that we can't be together live. But we are able to tell you a few things about us this year, and I see you can use the chat box to speak to each other. And if you have questions for us and for any of the presenters, please use the question and answer box. And after each presentation, Chari Cohen will answer questions, then Ju-Tao will answer questions. All have stepped away. This webinar is being recorded, as it's indicated, and we have, and I think we have a pretty good program for you. Some pretty interesting things to say tonight. But if I could have the next slide, you'll see the speakers tonight are Chari Cohen's going to give you a kind of a sneak preview update to the results of the patient conference that the foundation sponsored with the US FDA this summer, again done digitally, but had an incredible participation. I think it was more than 700 people actually live, and another something thousand were online, followed up by registration, or answered the questions. So that's really going to be interesting, what hepatitis B patients are thinking about with respect to treatments. And Chari Cohen, Senior Vice President of the Hepatitis B Foundation, will tell you about that. And then we thought it'd be neat if Ju-Tao Guo, the Baruch S. Blumberg Institute's Vice President for Research, told you about some updates of research at the foundation. This has been a very, very active year in the field. Good and bad things are happening. There have been some good things at the Blumberg Institute, created by the Hepatitis B Foundation, and as well. And we want you to know about that. If I could have the next slide. We're coming on to a very important year. We were founded in 1991 by Jan and Paul Whitty and Joan and I. And by the way, Joan is here. She's logged on as me, and I'm logged on as her. And so we're coming on 30 years. So this is going to be a year of celebration for us. And at the next year's AASLD, we'll have a whole year's summary and a reflection. And it's quite educational to to compare where we were in 1991 with where we are now, but still a lot to do. If we go to the next slide, please, Megan, you'll see that at the Hepatitis B Foundation's campus. So for those of you who haven't been here, and I hope everybody on the webinar does get a chance to visit us at some time. The Hepatitis B Foundation is on its own campus in suburban Philadelphia, in Doylestown, Pennsylvania. And the image you see is a drawing of the new building we're building. It's a 38,000 square foot expansion, $20 million expansion. It'll be our new laboratories for the Blumberg Institute, a new accelerator, as it's called, for the faculty spin-out companies. And we'll be in the middle of our current 14-acre campus at 120,000 square feet of labs and offices and instructional space right now. We currently occupy an old factory that we converted, and now we're going to have our first purpose-built building, and I hope you get a chance to see it. So this is a big year for us, and again, what a way to break into our 30th anniversary. Something else I want to tell you about, and maybe we're going to make this reception the place where we actually announce the winners of the Blumberg Prize. Every year the Hepatitis B Foundation names the winner of the Baruch S. Blumberg Prize, previously called the Distinguished Scientist Award. And And of course, we give that to an individual or individuals who have made outstanding contributions, transformational contributions in the area of hepatitis B virus research and liver cancer research. And, and the individuals that we get it are the VIPs in the field. The past recipients choose the winner along with our SAB panel. And this year I'm very proud and pleased to tell you that this year's recipient is Wenhu Li from the National Institute of Biologicals in Beijing, China. And Dr. Li is winning the prize this year. And normally the winner comes to the Hepatitis B Foundation, accepts his award or her award, and gives a seminar. This year we may have to do it digitally, but Dr. Li is receiving the award for the discovery of the hepatitis B virus receptor on hepatocytes. And that is a question that has eluded the field for decades. And the discovery of that a few years ago by Dr. Li and his colleagues is a seminal finding, and it really helps buttress discoveries in the field and also, of course, new therapeutics for hepatitis B. And there's already Merclid XB, which works by inhibiting the attachment of the virus to this receptor. So we're very excited about that. I think we're proposing we're going to use the AASLD reception as the place to announce the winner. This evening or tomorrow morning, the press release will go out. So congratulations to Dr. Li, and I know you'd all join me in extending those congratulations. If I can go to the next slide, please. I'll now introduce Chari Cohen, who is the Senior Vice President for the Hepatitis B Foundation and is a public health expert. And I don't have to tell you in this environment of COVID how good it's been to have an expert in public health on our campus. But she's going to tell you today about the results of the conference that the Hepatitis B Foundation hosted with the US FDA this summer in Beltsville, Maryland. And now I'll turn it over to you, Chari. Again, if you have questions, please put them in the Q&A box, and Chari will answer her questions right after. And Chari will introduce Ju-Tao Guo, who will then take questions. This is the last you'll see of me for the evening. I regret that because, again, I wish I could be there shaking your hands and having a drink together. But until next year, and welcome to the AASLD.
Thanks very much, Tim, and thank you everyone for coming. I echo Tim's sentiments. I wish we were in the room drinking, cheap wine and cheese and crackers. So hopefully you have a glass of wine and maybe some appetizers. And I will say, you know who your friends are when they come to a reception without any food or drink. So thank you for coming tonight. I'm really excited to talk about the work we've done over the last year. For those of you who were at our AASLD reception last year, we actually announced that we were going to be hosting the externally led patient-focused drug development meeting. So I'm really pleased to be sharing some initial results this year. I'm going to be talking about a couple of different efforts that we have completed over the last year that we call our patient engagement program, really making sure that the voice of the patient is heard, especially now. We all know we're at an important time in terms of drug development and clinical trial development, and it is critical that we document the voices of the patient. Next slide, please, Megan. Thank you. So these are the questions that we're trying to answer, and this is what has been guiding us. We want to find out and truly rigorously document what is the impact of living with chronic hepatitis B, physically, emotionally, professionally, and socially, and focusing quite a bit on people's experiences with stigma and discrimination around the world. And of course, we want to find out and talk about what patient experiences are with current treatments and what they want and need in terms of future treatment and future clinical trials. If we're going to improve clinical trial participation and diversity into the future, how do we do it? Next slide. So we had a bunch of different methodologies here. So one, we did host the first ever externally led patient-focused drug development meeting. So our Ph.D. was hosted and approved by the FDA. We had it in June, of course, virtually. And we did have over 650 participants. We've had over 3,000 people watch it since then. Pleased to say that the report from this meeting is now live and on our website. We just launched it 2 weeks ago, and it gave us a great insight into what people go through. We also conducted an online survey, had almost 2,000 people internationally and in the US complete that survey this year. We conducted 24 in-depth telephone interviews, so qualitative interviews with people living with Hep B in the US. And we completed a data mining and a qualitative analysis of about 10,000 patient email consults from the past 2 years. So we are working to analyze the data and publish. In fact, we've got 2 articles in the works already, and one being submitted very, very soon. But also, outside of publishing in peer-reviewed journal articles, we want to make sure that the information gets to our colleagues, patients, advocates, and people around the world who need it. So we're going to be sort of blasting this information out to the public as well. Next slide. So I'm going to talk a little bit today about some of the things that we have found and that we're starting to find. And again, this is just, we've started mining the data, but we have a lot of work to do. But just some overview. First off, from the Ph.D, which again, I was so pleased that we were able to do that. We did find and learned that there were significant physical and emotional impact for people around the world living with hepatitis B. And some of the major themes people focused on, not only so physical symptoms, fatigue was number one, but also pain, and that was also talked about quite a bit. But there was a lot of talk about people feeling isolated, people feeling self-stigma and stigma from others, and also people fearing the future, people waking up every day and being scared about dying young or developing liver cancer. And these are the kinds of things we don't always hear about, we don't always talk about, and how they affect people's relationships, their professional relationships and their familial relationships as well. People also documented current treatment challenges, especially cost and access in the US and around the world. But also we heard quite a lot about the struggle of taking a daily pill for many, many years. And this is something I think, again, is really important for people to hear, how difficult it is to take a pill every day. Sometimes it was a matter of not having continual access, and sometimes it was a matter of just the emotional impact and the reminder every day of taking that pill. Of course, we saw a strong desire for a loss of surface antigen and reduced risk of liver cancer. And then among those who have never been on treatment and aren't on treatment, there was quite a lot of talk about how they feel like they're not doing anything and that they're unable to do anything to help their future and to feel healthy outside of things like maybe herbal supplements or staying healthy with diet and exercise, but people are still scared. Next slide. People, so this is data from our survey that we used, and we're kind of triangulating all the data together to get a big overview, a big picture. And so what we saw here, we asked people to report their medical challenges that they have living with chronic hepatitis B. You can see both internationally, which is in the yellow, and US, people are facing quite a lot of challenges. So of course, internationally, we see more people seem to be facing challenges, but they're related to finding a doctor who's knowledgeable about Hep B, and then of course, cost of the testing, especially every 6 months. But you know, even in the US, it's not trivial. We have over 40% who are facing these challenges, and that's something that I think we need to address. Next slide. In terms of future treatment, we asked people both in the survey and the interviews and during the PFDD what they want for the future. Of course, I think what we saw were most people preferring oral treatment, although there did seem to be some acceptability for the future for injectables or infusions. And you know, we saw willingness to participate in clinical trials. However, there were quite a few caveats. I think there were people who, you know, overall, we saw fears about safety, people who wanted to maybe learn more about clinical trials, but wanted to make sure that they knew what they were going to be taking, that they could see a safety profile for whatever drug they might be taking. And of course, people wanted to know that they weren't going to be worse off for joining the clinical trial. We saw this especially for young adults, people with young families, and also for older adults, people who were saying, you know, I might be interested in joining a clinical trial, but maybe I'm just going to leave well enough alone and be a little bit more cautious. And then of course, people wanted to know that if they're going to join a clinical trial, that they would want a higher chance of functional cure. And of course, we saw international versus US differences, and we're going to talk about that in a little bit. Next slide, please. So we also asked people to list what their #1 most important outcome would be for future treatment. You know, what is it that they want more than anything else? So if you look at the yellow bar, you'll see internationally, the #1 considered to, and this is from the survey again, what people consider to be the most important future treatment internationally is loss of surface antigen. And this is actually not surprising when we look at the email consults that we get and the phone consults that we get. Internationally, people are being discriminated against because they have surface antigen, right? They can't get jobs, they can't immigrate. So it's not surprising to see that outside the US, people want to lose surface antigen. And of course, also want to be able to stop taking their meds after 6 or 12 months because they're having access issues and cost issues. In the US, you can see that the #1 most important factor was decreased risk of liver cancer. And again, you know, what we were hearing from the Ph.D. is that this unending fear of liver cancer is kind of driving a lot of the emotional impact. So not surprised to see that either. Next slide, please. In terms of willingness to join a clinical trial for a future potential functional cure, this is interesting. So you could see overall, international survey respondents were more likely to join a to consider joining a future clinical trial than US folks. And again, you can see, you know, in both cases, the bar goes up as the chance of functional cure goes up. But in the US, you know, it's certainly lagging until you hit the 90% chance of a functional cure. But I think it's something to remember. As we know, clinical trials are being developed around the world. One thing to remember is that both for our PFDD and for the survey, a large percentage of people who participated were from Africa. And so a lot of, you know, what we hear in the consults is that people in Africa are desperate for a cure, desperate for treatment, desperate for being medically managed. And so I think it's something we need to remember as we're thinking about who we're going to be focusing on for clinical trials in the future. Next slide. So that's it for my section. We're going to be throwing a lot more information out there. You can find the report now on our website. And we will be sharing more information on the data from the survey, the PFDD, and the focus groups and the interviews. But I wanted to make you aware of a couple of new things that have just recently come down the pike at the Hepatitis B Foundation. On the left, you can see a new report that we just put out to coincide with open enrollment for health insurance. As you know, people are facing potentially very high prices of hepatitis B medication in the US, and we're seeing some discriminatory drug tiering. We did an analysis of a couple hundred of health insurance companies across the US, and we created this tool for people living with Hep B to help them find appropriate health insurance packages that won't break the bank, hopefully, for hepatitis B drugs. So if you're a clinician or you work with patients, please feel free to share that with them during open enrollment now, especially. In the middle, you can see our new liver cancer drug watch. I know you're all familiar with our Hep B drug watch. Our HBV drug watch has been up since before 2010. I'm very, very pleased. We worked on the liver cancer drug watch for quite a while, and we just made that live last week. So please go in and take a look, and we'll be updating that regularly. And then on the right side, and this is something I'm really excited about, as you know, the foundation played an important role in making sure that people who have hepatitis B are not discriminated against in the US. And of course, as you probably still know, we do get medical providers, healthcare students contacting us almost on a weekly basis who are facing discrimination. So along with some of our our colleagues and advocates, we did speak with HHS this year in the very beginning. In fact, it was the last meeting I attended in DC before lockdown. And we were lucky enough to meet with Admiral Drar and talked about the discrimination that was happening, and they were very committed to doing something about it. So as you'll see on the right-hand side, Admiral Drar and Roger Severino, who is the director for the Office of Civil Rights at HHS, they have released a new letter for schools, healthcare schools, reiterating that people with hepatitis B cannot be discriminated against, and that and so they will be sending this letter to schools across the US, and we will be sharing it widely as well. But it's one more tool that we now have that we can use to help students and healthcare providers who are facing discrimination because of their Hep B. And with that, I am now going to turn it over to my colleague, Dr. Ju-Tao Guo, Vice President of Research at the Baruch S. Blumberg Institute. Oh wait, I think I'm supposed to do questions first. Before I turn it over, let me just check the Q&A. I don't actually see any. If you have any questions about any of the data that I shared, yeah, I don't see any. So please feel free to keep putting questions in the chat box and we can answer them all at the end. But now I'm going to turn it over to Dr. Guo. Thank you.
Oh, sorry, I just muted. So good evening. It's a great opportunity for me to introduce a research program in the Blumberg Institute. So our institute that was founded by the government grants and also through the collaboration with Builders and also the donors from the different place. So our research program mainly including two parts. One is about the biological marker discovery for the early detection of the cancer. And another one is antiviral drug discovery and development. So today, I mainly focus on introduce to you our research progress on the antiviral drug discovery and development program. Next slide. So here is a current status quo. So in the best case scenario, that was combination therapy of the antiviral drug nukes like tenofovir or entecavir in combination with pegylated interferon. That's after 1.5 year treatment. So surface antigen loss is about 9%. So next slide. So then people would ask that the hepatitis B, that was the first discovered hepatitis. So now it's more than 50 years. We still don't have a cure. However, for the hepatitis C, that's after 25 years of this discovery of the virus, so curiosity therapy is available. So why is that? That's because it's two totally different viruses. So hepatitis C virus, that's an RNA virus, that's by suppression of the viral replication, you can actually completely eradicate the virus from the body and to achieve the virological cure. Next slide. For the hepatitis B virus, so the real challenge is that we cannot eliminate all the infected cells or cure all the infected cells. And that is mainly because viral DNA in the form we call the cccDNA that can persist in the infected cell, the nuclear, for a long time. So even the immunological resolution of the present infection that maybe is most likely is not a complete eradication of the virus. That actually is immunosurveillance of the residual infection or latent virus infection. So that's a reality is that we may never able to completely achieve a virological cure. So next slide. The goal of this new therapy that as team and have led us to published this opinion paper 7 years ago in antiviral research indicates that the goal should be achieve surface antigen loss, or at best, the surface seroconversion. Oh, next slide, please. Seroconversion with affinity therapy. So, of course, the ideal scenario should be the seroconversion into the surface antibody. But that's, in most cases, that bar maybe is too high. So reality is loss of the surface antigen from the circulation. So next slide. So consider, the reason for the current antiviral therapy like polymerase inhibitor, or called nukes, that cannot cure, achieve the functional cure. We consider that the major reason is that one is may not completely suppress viral replication. And another reason is cannot restore a functional immune response, because the antiviral dysfunctional immune response, or T cell or B cell exhaustion, that's most likely is driven by the sustained existing of the large amount of viral antigen. So in between these two mechanisms, we think that reduce the virus antigen load is a key for the restoration of the functional immune response. So based on this mechanism, next slide, we propose that for achieve a functional cure should be through the 3 consecutive stages of the treatment. The first stage, the goal is complete inhibition HBV replication. That can be achieved with nuke in combination with new HBV replication inhibitors such as capsid assembly modulators. And after this, that's a small fraction of patients that have a higher level antiviral immune response. They may able to achieve loss of the surface antigen, and but most people will not. So then we need to reduce the viral antigen load through targeting cccDNA or targeting viral RNA through the antisense oligos or the sRNA or other mechanism. And finally, you can activate the functional immune response through the therapeutic immunization or other immunotherapy approaches. So I think the immunotherapy, the stage must build on the first 2 stages. When the viral replication is high, if you start the immunotherapy, that may not able to activate, or after activation, you may induce a severe liver damage. So next slide. So based on all these considerations, so currently in the field, there's several approach for the antiviral drug development, and have the continued development of the drugs targets polymerase, like polymerase activity itself or RNase H activity, and also targets the capsid assembly and the disassembly, and several approach target viral RNA stability, and also have a several approach for the entry, inhibit virus entry. So next slide. So here, right now in a clinical trial, there's about more than 10 compounds in clinical evaluation for of the capsid assembly modulators for their ability to treat the chronic hepatitis B. So all these we call the core protein allosteric modulators and others also called capsid assembly modulators. So those compounds can bind to the core protein dimer-dimer interface and misdirect capsid core protein to assemble into either no capsid polymer, and that is form aggregates in cells and subsequently degraded. That's called type 1 CPAM. And the type 2 CPAM, through binding to the same pockets and induce the empty capsid formation, so that cannot package the viral pgRNA and then prevented viral DNA replication. So, and also both the type 1 and the type 2 CPAMs, that's in addition to inhibit pgRNA encapsulation and the DNA synthesis, they can also, next slide, they can also induce the mature rcDNA containing capsid disassembly. So these effects actually is a basis for those compounds can inhibit the de novo cccDNA synthesis. So those class of compounds you see have a dual effects, inhibit DNA replication and new cccDNA synthesis, right? And recently, next slide. So recently we published a review article to summarize the current status of the Zeus class of compounds developments. And generally speaking, they demonstrate a very nice and potent antiviral activity. And in combination of the nukes, they can achieve much more complete viral replication inhibition. And in news, recent a few days ago, that's the assembly that's capsid inhibitor in combination with nukes that's not able to achieve a sustained viral response. So I think that in principle, that is not something unanticipated. It's actually, based on the previous, the few slides ago, I introduced that, you simply by inhibiting viral replication, that you may not achieve surface antigen loss or sustained antiviral response in most majority of patients. So that is not a surprise. However, so the capsid inhibitors that will be key components in the antiviral functional cure of the HBV therapy. Next slide. So another project that was led by Dr. Tianlong Zhou in our institute, that's in collaboration with Abilis Biopharm. So work on a compound called RG7834. So this compound that's discovered by Roche that can induce the degradation of the viral RNA. And Tianlong have found that compounds induced viral RNA degradation is dependent on these RNA elements we call the PRE or this stem loop structure. And he did very detailed studies to find that as long as you mutate 4 nucleotides on the bulge of the stem loop, you can totally make these compounds not effective. And later on, that's studies from Roche and Novartis, and found that's very interesting. And a cellular protein alpha called ZCCHC14 is the RNA helix combining to this stem loop and recruits this protein is a non-canonic poly-A polymerase binding to this stem loop. And once the viral RNA degraded through the 3' and lose the poly-A tail, and this polymerase can actually add the poly-A tail to stabilize this viral RNA. So this compounds is this non-canonic polypolymerase inhibitor. So next slide. Tianlong have developed a very nice in vitro enzymatic assay and clearly shown that those class of compounds that you see dose dependently inhibit this RNA polymerase activity. So next slide. So as we have said that the cccDNA is one of the big barrier towards the functional cure. So in my lab, we have been have a long interest in to understand how the cccDNA was made, how that transcription is regulated, if we have any chance to eliminate cccDNA, or if we cannot, we just can silence its transcription. So during the last 15 years work, we have been made several contributions to the cccDNA biosynthesis studies. For example, we found that from the rcDNA to cccDNA, that involved the one of the intermediates to remove the protein we called deproteinize rcDNA, and then another group that Jianming Hu's group found there is another intermediate between this dprcDNA to cccDNA. So then we identify the topoisomerase 1, topoisomerase 2, alpha delta that was actually involved in this cccDNA synthesis. And this is picture is I just take from one the commentary to the recent paper that was described, published by Princeton University. A group from Princeton University described the in vitro cccDNA synthesis, and they was able to establish a biochemical system to use 5 protein to synthesize cccDNA. Actually, very interestingly, and the components 3 is overlapped, and these 2 systems together. So now I think that we get a much more complete picture about cccDNA synthesis molecular pathway. Of course, all these components, that is very important for host cell function. So as no one seems is a good target for antiviral therapy. Next slide. So then next question is, can we get rid of it or silence it? So the study from my own lab that found, the cccDNA is very hard to get rid of it, is that you can only dilute during the cell division. And however, things like interferon, if you treat the cells that can actually inhibit the cccDNA transcription, and especially we use DHBV cccDNA as a model, we found that if you treat the cells with interferon for just 1 or 2 days, and then you remove it, that's interferon-induced cccDNA transcription can be maintained for a long time. So our previous study found that that's actually induced a cccDNA associated histone modification. And in the last few years, we have did a lot of the work to try to identify the interferon-inducible cellular genes that mediates these epigenetic changes. So recently we published these results and found that SMAC HD1 is a chromatin epigenetic modulator protein, and also the PML body components that seems induced by interferon and can recruit to cccDNA and suppress its transcription. And this information may lead us try to learn from how interferon works and to devise this assays for the discovery of the compounds that can inhibit the cccDNA transcription. So finally, we also want to, next slide, work on the, discover a small molecule that can activate the host immune response. So one thing we are focused is on the discovery of the STING agonist. So through in-house screening, we discovered two human STING-specific agonists, and those agonists have three effects. First, if you treat the hepatocytes with this STING agonist, they can actually inhibit the cccDNA transcription, reduce pgRNA and other viral RNA, and suppress viral replication. And also, those agonists can also work on the macrophage and other cells and induce cytokines to indirectly inhibit viral replication. And a third function, we found that they can actually stimulate the NPC or the antigen presentation cells and present antigen presentation to the naive T cells. So we believe that, again, for the achieve the functional cure of the HBV, indeed need a coordinate efforts through the inhibitor viral replication, reduce virus antigen loads, and then activate the viral immune response. So here, this is just an outline of our major research program. So next slide. Here is our team of the investigators, except myself. Dr. Jinghong Chang is a virologist and mainly responsible for the antiviral drug screening program. And Dr. Zhu, I just mentioned that was work on the, this RNA reducer compounds mechanism. And Dr. Zhu is a medicinal chemist that help us to do all this lead optimization. And John Kopp is a computation chemist and help us do all this structure modeling and docking. And Dr. Patrick Lam is the distinguished professor of our institute, is very senior medicinal chemistry chemist and provide a lot of consultation for our antiviral drug development program. Thank you. I will take a question here.
Great, thank you, Ju-Tao. I'm looking, I don't see any questions, but there's still, so if you go into the Q&A box or even the chat box, oh, we do have a question. Okay, Ju-Tao, here's the first one. Nucleocapsids appear to be disassembled at the nucleopore and then reassembled in the nucleus. What cellular mechanisms are responsible for this, and what would prevent it from occurring again with CPAMs?
So I think that, for kinetically, for the type 2 CPAP, that seems assembly, the capsid assembly, that's right way in the cytoplasmic. So long-term treatment, that's usually induce accumulation of the empty capsid in the cytoplasmic. However, for the type 1 CPAP, that's actually, induce the reassemble of the core protein dimer in the nuclear, not into the empty capsid, actually into the no capsid aggregates. That's actually can accumulate in the nuclear, especially for the no dividing cells. So we don't know what's the biological consequence yet.
Great, thank you. So I do see another question from Dr. Gesh asking about Roche potentially pulling interferon or PEG interferon from the market. I don't know anything about that. I don't know, Ju-Tao, have you heard anything?
Say again?
A question about Roche potentially pulling PEG interferon or interferon from the market.
Oh,
I have not heard.
yeah, I didn't hear that. So actually, I think that despite the poor tolerability, that's in terms of functional cure right now, either the increased nuke cure rates, or I think that's another miracle I actually not mentioned, that was that's replicas, that's NAP, right? That seems it's alone is not work, but you need a combination with interferon and the nuke. That triple combination seems can achieve almost about 50% of functional cure. That is amazing. Unfortunately, that's how exactly that things works is not known. Oh, I see Peter have a question here. Hi,
Yeah.
Peter. So I think that right now for the it will be great if the nuke and the CPAP can be in a combination. So that's, in my point of view, that maybe can achieve longer, more safer and long-term suppression of the viral replication. And even if that's not achieve a functional cure by that combination, I do hope, because now we have more cards to play, right? So we can actually start treatments early for most of the chronic carriers, because I also just heard that when the patient after age 50, at that point, even you lose surface antigen or achieve a functional cure, that's the HCC incidence may be not dramatically reduced. So that means the early treatment is very important. But right now, if we only have 2 nukes that's available, so that maybe we cannot start treat too early because the option is limited. If now we have more option, and so I think that we can actually consider to expand the treatment guideline to including more patients to be treated.
Thank you, Ju-Tao. I just want to note, so Dr. Pirillo put in a comment in response to the Roche question, basically stating that pulling interferon would be a mistake. So he states not only is it the only agent that's been clearly shown to induce functional cure, but it continues to be used in combination and will likely continue to be used in combination with new therapies. So yeah, we can continue looking at this and just maybe do some research and find out if that might be an option.
Yeah, I don't know what's their reason. So what I heard that the Roche also seems stops their HBV program in Shanghai.
Oh.
Yeah.
That's not good news. Yeah, maybe we should do a little digging into this and see if that's actually happening. Yes, and I agree. So Peter Revel also just said another key will be a finite therapy, so a daily pill does not have to be taken for life. That I completely agree with that. It's something that we hear quite a lot. We heard through the Ph.D. and the interviews and surveys, and of course through our patient consults. It's taking that daily pill for many, many years is really difficult. I think also from an access and a cost issue as well. What we're seeing from a lot of patient consults in other countries now is that, especially in lower resource countries where maybe they're being managed by doctors who aren't as familiar with managing and treating Hep B, people are being given 6 months of tenofovir and then told that after 6 months they can stop taking it. And so we actually see that that kind of question come in quite often. So having an actual finite therapy would be really helpful. Agreed. Let's see. There's one more question that came in. With the Biden presidency, how can we all unite to eliminate liver cancer? Oh, Lauren, thank you for asking that question. We were just talking about this yesterday. I'm hoping that the cancer moonshot is going to once again become a priority under President Biden. That's nice. Yeah, so hoping the cancer moonshot and so liver cancer was a priority originally, and we know that there were some liver cancer specialists who are a member of the Moonshot Task Force, and we are hoping, and again, I think us as advocates, the advocates in the room, we are really good at coming together. I think one of the things we can do is what we do best, and that's make our voice heard and talk about the importance of prioritizing liver cancer. And ask them what their plans are. So Lauren, we should definitely follow up. I think we're already pulling together language for that. And I'm hoping that the deadliest cancers are going to become priority over the next couple of years. I'm optimistic. Thank you. Great question. Just checking to see if there. I don't see any other questions. We can give it just another few seconds. But thank you for all the questions that are coming in. It's great to have this kind of conversation, even without the wine. All right, well, I don't see anything else coming in. So with that, I just want to thank everyone for attending and close us out for the evening. I know we've got, Friday, Saturday, Sunday, we've got 4 jam-packed days at AASLD. I myself am gonna have to have 2 computers open because I have 2 sessions, there's like 2 sessions I have to go to at the same time. So everybody please enjoy AASLD and thank you for coming tonight. And hopefully next year we can be together in the same room again. Good night.
Thank you all. Thank you.
Thank you.
Take care.
Bye-bye. See you next year.